Hello friends, I am Gaurang Makwana. I welcome you all to my another video tutorial Biotech guide. In this video lecture, we are going to discuss about RNA processing, also defining as a post-transcriptional modification or a co-transcription modification. We'll brief about steps which are involved in RNA processing. First is capping, then polyadenylation and RNA splicing. So today's video going on very interesting level. Stay tuned with us and enjoying this video lecture. Let's start first introduction. We know central dogma where DNA converts into the mRNA and then mRNA converts into the protein. During conversion of a DNA into mRNA is defining as a transcription process. So after transcription we have a pre-mRNA, not mature form of RNA, which will be further going for a translation process. So here what is pre-mRNA? So pre-mRNA is defining as some parts of an intron and some parts of an exon is there. Here intron is the non-coding part and exon is the coding part. So here you can see this kind of a structure is there for a pre-mRNA. But for the translation process we required a mature form of RNA. That means only the exon part is there, not non-coding intron part is there. So for that we required one process that is known as a RNA processing. So in this video lecture we are going to brief about this process, RNA processing. In prokaryotes no RNA processing is necessary, but in eukaryotes RNA processing is required. Why? Because after transcription in eukaryotic organism we have highly unstable and heterogeneous mRNA, also defining as a hnRNA, which will be further going for a highly stable and a compact mRNA through a process is defining as a RNA processing. This process is occur after transcription, that's why is defining as a post-transcriptional modification or a co-transcription modification. As we discussed earlier, we required mature RNA. So what is the perfect structure of a mature RNA? Here you can see this is the mature mRNA structure, where you can observe at the 5' side cap is also there, and the 3' side poly tail is also there, and the coding region is also there. So we required this kind of a mature mRNA. So for that we required some process, capping, polyadenylation and RNA splicing. So we'll discuss brief in this video. First, capping. So this is the normal basic structure of a RNA, where you can observe this is the base, then this is the ribose sugar, and this is the phosphate molecule. Okay, so here stability is required at the 5' side, also defining as a cap side. So 7-MEG molecule is attached at the 5' side by the use of a guanyl transferase enzyme, which provides stability at the 5' side. Okay, and 5' to 5' triphosphate linkage is formed between a mRNA and a 7-MEG molecule. Same is happen at the 3' end, but here poly tail is added at the 3' side by the use of a PAP poly A polymerase enzyme, which provides stability at the 3' end. So here you can see this is the process which occur in a polyadenylation process, and this kind of a tail is joined at the 3' end. Now most important RNA splicing is happen after polyadenylation process, where introns are removed. Introns is non-coding region and exons are joined together, which is defining as a coding region. Okay, and here another structure is formed, which is defining as a spliceosome complex formation take place. This spliceosome complex contain snRNA, small nuclear RNA, protein and snRNPs. Here snRNPs plays a major role for the cleavage and the joining of an exon. These RNPs are basically four main types: major, minor, small nuclear and a small causal body RNPs. For this RNA splicing or a spliceosome complex formation, major snRNPs are involved. These are U1, U2, U4, U5 and U6. So let us discuss brief. Before that, let's revise mature mRNA structure. At the 5' end, guanine and uracil base is there. At the 3' end, adenine and guanine base is there. And intron part is there which contain adenine residue branch. We required a mature RNA, that means only exon part. So for that some steps are involved for the cleavage. Step 1, UN snRNP bind at 5' site. U2 snRNP bind at adenine residue. As we discussed, intron part contain adenine residue, so at that part U2 snRNPs are binds. Then primer of a U4, U5 and a U6 binds at a 3' splice site intron, and in this step spliceosome complex take place. You can see here. Then after loop formation is occur and conformational change occur in this place. Then after a cut is formed at the 5' splice site and a structure is formed which is defining as a lariat structure. Then step 6, U1 and U4 snRNPs are released, and U5 and U6 snRNPs change its position. Then at the 7th step, 3' splice site cutting is there or cleavage is happen, and at the end, step 8, lariat structure is now released and exons are joined together. So this is the RNA splicing process. Alternative splicing. So what happen when pre-mRNA is having a large number of exon coding region? So that time alternative splicing take place where splicing is happen in this pre-mRNA conversion into the mRNA, mature form of mRNA, and that time many kinds of mature mRNA is generated. So basically that allows for the production of many different kinds of protein. So a single RNA can by combining different exons during a splicing create many different protein coding messages through alternative splicing. We and other eukaryotes can sneakily encode more different proteins than we have genes in our DNA. So basically at that time of alternative splicing we have more different kinds of protein than we have genes in our DNA. After protein coding message have been processed by capping, splicing and addition of a poly A tail, they are transported out of the nucleus and be translated into the cytoplasm. So this is the alternative splicing. Now importance of RNA processing. So RNA processing is required to control a low genes expression, then also required to create families of a related protein and affects the fate of a resulting mRNA. So these are the importance of RNA processing. Here we completed our video. I hope you enjoying very well and got it very well. Thanks for watching, and if you want to get more and more interesting video regarding like this, join with us and be a perfect biotechnologist. Thank you so much friends.